if we look at water regulation in the human body, what we notice is this. Let's take a look at table 14-3, and we see that most of the water that comes into a human body comes in, not surprisingly, via liquids, but a surprisingly large amount in an average person in an average day makes its way in via food. And so we see that 1,100 mils per day makes its way into the body via food. And then, in my opinion, kind of a surprisingly large amount is the result of metabolic production. And so 350 mils, although it's certainly by far the least, but is made per day in the body via metabolism. So that makes a total of 2,850 mils that comes in each day. So in an average day, then, what we see is that about 900 mils of that is being lost via these insensible mechanisms. In other words, mechanisms that we don't actively sense, and so the lungs are part of that, and the skin, ever so slight sweat via the skin is another part of that. So the next category we have for loss is sweat, which would, of course, vary quite a bit on a daily basis depending on the outside temperature, humidity, and activity. But 50 mils is used as an average in this particular example. Another 100 mils is lost in feces, and by far the most is lost in urine, with 1,800 mils out of the 2,850 that's lost each day. So it is the responsibility of the renal system, the topic of this chapter, to make sure that that intake and outtake is balanced. And so the renal system, as we're going to talk about in the remainder of our chapter here, has a tremendous ability to either excrete additional water, releasing it from the body if there's lots of water taken into the body, or release less water up to a point. There is a limit, but up to a point, release less water and therefore conserve water if there's a deficit of water coming into the body. Lastly, we look at salt regulation, and so what we see is this, that in an average day, an average person takes in about 8.5 grams of salt via their food, although that can, of course, be modified greatly by carefully selected diet. And then the output there is a very small fourth of a gram in sweat, fourth of a gram in feces, and then most of that is lost via the urine. That intake and output, of course, in the case of salt, must also be perfectly balanced. And so, once again, it is the responsibility of the renal system to balance the intake and outtake of salt, as we'll see over the next couple sections. What we see is that both sodium and water are freely filtered, and then they are almost entirely reabsorbed, 99% reabsorbed, and they're not secreted at all. And what we also see is that both sodium and water reabsorption can be modified, as we mentioned previously, to adjust the overall concentration of both sodium and water in the body. Most of this reabsorption occurs in the proximal convoluted tubule. However, this reabsorption is not adjustable. It does play some other important roles physiologically, but the hormonally adjustable portions of both sodium and water occur in the distal convoluted tubule and then in the collecting ducts. Let's go down and look at what sodium and water reabsorption, actually just sodium in this case, reabsorption looks like in both the proximal convoluted tubule and then later in the collecting ducts. And it varies, the way that it occurs varies depending on the part of the tubule in which you find yourself. So let's begin here in the proximal convoluted tubule. The common denominator that we find in both the proximal convoluted tubule and the collecting duct is a sodium-potassium pump that's found on the basolateral side of the epithelial cell of the lumen. And so this particular pump is, of course, pumping sodium out into the interstitial fluid and trading that for potassium ions, which are transported in to the cytosol of the epithelial cell. But we also notice this, so actually maybe we should slow down here. So we're pumping sodium out, and so the small sodium symbol here indicates a low concentration of sodium found in the cytosol of the epithelial cell. That then drives the movement of sodium down its concentration gradient from the tubule of the lumen into the cytosol of the epithelial cell. And two things are accomplished as that sodium moves into the cytosol. One is that organic compounds, such as glucose, are moved from the lumen of the tubule into the cytosol. And so the small x here, for instance, X is representing some sort of organic compound, let's say glucose in this case, and so there's a low concentration of glucose in the tubule, but a high concentration in the cytosol, and that's, of course, active transport that is driven by the movement of sodium down its concentration gradient, a form of cotransport. And then down here we have countertransport, and so sodium, the movement of sodium in is responsible for the movement of hydrogen ions out of the cytosol into the lumen of the tubule and then eventually out of the body. Again, this is in the proximal convoluted tubule. If we make our way down to the collecting duct, what we notice is this, that the whole situation is simpler. Once again, as we said a moment ago, we have the sodium-potassium ATPase located on the basolateral side, but in this case we simply have channels, protein channels that allow sodium to diffuse into the cytosol. In this case, we don't have this cotransport or countertransport like we did back in the proximal convoluted tubule. If we look at water, the reabsorption of water, we see, not surprisingly, that water follows the sodium gradient as a result of osmosis. So step number one here is the movement of sodium, as we just talked about, into the cytosol, and then of course sodium is being pumped out here, and then eventually making its way into the peritubular capillaries. And so this leads to a low osmolarity in the tubule of the lumen, and a higher osmolarity, of course, in the cytosol and in the interstitial fluid. Water follows as a result of osmosis. The water pressure builds up in the interstitial fluid here, and that then forces the water into the peritubular capillaries via bulk flow. So just like we see in the case of the digestive system, the reabsorption of water is driven by the movement of sodium via osmosis. Now, perhaps most importantly, in our discussion of the reabsorption of water in the renal system, we notice that the renal system can vary the reabsorption of water quite a bit to make a very concentrated urine or urine that is much less concentrated. And so this adjustment, as we mentioned previously, is done in the collecting ducts. And the collecting ducts are permeable to water, but their permeability depends primarily upon aquaporins, these protein channels that are permeable to water and allow water to move across the plasma membrane. The proximal convoluted tubule is always very permeable to water, and so it always lets sodium and water come through in equal amounts, but the permeability of both the cortical and the medullary collecting ducts vary widely. So the collecting ducts vary as a result of the insertion of aquaporins into the luminal membrane of the epithelial cells of the tubule. And so that is stimulated, as you would imagine, by vasopressin, also known as antidiuretic hormone, which binds to its receptor, activating an adenylate cyclase, which then raises levels of cyclic AMP in the cell, which activates a protein known as protein kinase A, which then phosphorylates proteins that are responsible for the fusion of vesicles containing aquaporin 2 with the luminal membrane of this epithelial cell. And so what we notice is that this makes the luminal membrane much more permeable to water, and so a great deal of water then moves into the cytosol and then out of the cytosol via the basolateral membrane through other types of aquaporins, aquaporin 3 and aquaporin 4. These aquaporins are not regulated by vasopressin, but they're always present. But it is the insertion of aquaporin 2 that allows us to regulate the concentration of our urine. Under high ADH levels, then what we see is that less than 1% of the filtered water is actually excreted. In other words, if this luminal membrane has these additional aquaporins, more water is moving out of the lumen of the tubule into the cytosol, into the interstitial fluid, and so very little water is left in the tubule to actually become urine. On the other hand, if there's not much vasopressin present, then a tremendous amount of water moves past here, and there's a great amount of urine that's excreted from the body. We now need to look at the processes that allow humans and other mammals to make concentrated urine. The creation of concentrated urine is important because it allows organisms to live independently of the water. If we look at amphibians, like the mud salamander that's present on this screen, we see that they have kidneys that have very little ability to concentrate urine. It's just not adaptive for them since they live in the water. There's no reason they shouldn't drink water, a large amount of water, and excrete a large amount of water. Humans, on the other hand, have the ability to concentrate their urine down to about 1,400 milliosmoles. So our the plasma osmolarity is about 300, and so this means that we can produce urine that's approximately 5 times more concentrated, approaching 5 times more concentrated than the concentration of our plasma. What we also notice is this, that every day an average-sized human produces about 600 milliosmoles of waste. And so this means that if we can concentrate our urine down to 1,400 milliosmoles, if we divide 600 by 1,400, that means that we've got to excrete about 450 milliliters per day. And that's known as the obligatory water loss, or sometimes the minimum excretory volume. So that's just the amount of waste we have, and we have to dilute it in something, and so that's as concentrated as we can get. Therefore, we're releasing about that 450 mils per day. So the question is, how do we do that? And the answer lies in the loop of Henle that we talked about previously. That's part, of course, of the nephron tubule. And we need to take a look at the loop of Henle here. Here we see the descending limb, and here we see the ascending limb of the loop of Henle. And we see several numbers here. The numbers represent the osmolarity that's found in first the descending limb, and then the ascending limb, and then these numbers out here, of course, represent the osmolarity that's found in the interstitial fluid. Now, the way that this works is the following. The ascending limb of the loop of Henle is impermeable to water and impermeable to sodium, aside from pumps that are pumping sodium out of the filtrate and into the interstitial fluid. And so we begin with that. So imagine the water starting up here, and as the water starts up here, then, or the filtrate starts up here, then the sodium is pumped out and pumped out and pumped out and pumped out. And so that's why we have this drop in the overall osmolarity that goes from 1,000 to 700 to 400 to eventually 100, which is, of course, hypoosmotic relative to plasma as we reach the ascending, the end of the ascending limb of the loop of Henle. So we're pumping salt out of the ascending limb. This is creating this hyperosmotic environment in the interstitial fluid that we see here. And what we notice is that this is drawing water out of the descending limb of the loop of Henle. And so we go from 300 to 600 to 900 to 1,400. And so by the time we get down to the tip of the loop of Henle, then we have an extremely concentrated filtrate. Now, I believe that we've already specified this, but just to make it clear, the descending limb of the loop of Henle is obviously permeable to water. Otherwise, none of this would work. So water's leaving here, as these arrows indicate, and the filtrate, the ultrafiltrate, is becoming more and more concentrated as we make our way down. This means that we have a tremendous concentration of solute and much salt available to start this pumping as we make our way back up the ascending limb of the loop of Henle. So the importance of all this, you may wonder, well, what's the importance? We go from 300 to 1,400 and then all the way back down to 100. But what's important is this, that we maintain a high interstitial osmolarity in the deeper layers of the medulla of the kidney. And so this is in these medullary nephrons that have a loop of Henle that dips way down into the medulla of the kidney. 15% of the nephrons are medullary nephrons, as we mentioned earlier. So the importance is that this 1,400 milliosmolar concentration is what allows water to be drawn out of the collecting ducts when those aquaporin 2s are inserted. Otherwise, there's nothing about the aquaporins that's pumping water. So if you put the aquaporins in, water would just sit there if there wasn't this solute concentration gradient. So to summarize, it is the loops of Henle that allow us to establish this extracellular in the interstitial fluid concentration gradient. It is this concentration gradient that allows us to reabsorb water and excrete a urine that is more concentrated than our plasma. In this diagram, we get to see beyond the loop of Henle all the way through the distal convoluted tubule and the collecting ducts. So what we notice is this. We've already talked about the reabsorption of fluid as we go through the ascending limb, the descending limb, and then the ascending limb of the loop of Henle. And what we see in the distal convoluted tubule is this, that once again we have a very low permeability to water, but we're pumping sodium we're still pumping salt out of the lumen of the tubule. And so the osmolarity here actually drops down far below what we see in plasma in the distal convoluted tubule because of the action of pumping that salt out of the lumen. Then we make our way to the collecting duct. And so first, in the cortical collecting duct, we see that we're fairly permeable to water, and so permeable to water and to salt, and so we have this exodus of water primarily. That means the concentration of salt that remains in the lumen goes up and up and up till we're approximately isotonic to plasma at 300 milliosmoles. And then we head into the medullary collecting duct. And so finally at this point we can see the importance of the osmotic gradient that we created via the loop of Henle. So depending on how much antidiuretic hormone has been released, and therefore how many aquaporin 2s have been inserted into the luminal membrane of those epithelial cells, then if we have a lot of antidiuretic hormone released, we're going to end up with a filtrate at this point. And of course, with this filtrate, we can almost call it urine at this particular point of the story. It's going to be isotonic to the interstitial fluid. So that's as concentrated as we can get with our kidneys, 1400 milliosmolar. And the reason for that limitation is a result of the fact that we only achieve a 1400 milliosmolar concentration with our loop of Henle. We also see urea being recycled here. So urea is moving from the medullary collecting duct into the loop of Henle, and that also assists in maintaining this high osmolarity in this particular region of the nephron. A good question about this process would be, why don't we lose this concentration gradient as a result of the capillaries that are present in this particular area? And the answer is that we would if it wasn't for the nature of these capillaries. So what we see is this, that we refer to the capillaries that surround the loop of Henle as vasa recta, which are very simply peritubular capillaries that make that long loop down into the medulla and then back out along with the loop of Henle. And what happens in the vasa recta is the following. We form these so-called hairpin loops. And so the vasa recta go down, 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 following the loop of Henle, and then they go up, 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 back into the cortex. And as they go down, what we notice is that we have salt that's diffusing in and we have water that's diffusing out. And so ultimately, we're mirroring the concentration of the extracellular fluid as we go down, achieving a concentration of 1200, which is close to the 1400 that's achieved by the loop of Henle. And then as we go back up, we can see that the converse is happening. We're losing salt and we're gaining water. We go up, 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 and up. And so the end of the story is this, that those capillaries are so permeable that they let salt water in on the descending limb of the vasa recta and then out on the ascending limb. And so they are transporting oxygen and nutrients and some waste products, but they're not disturbing this osmotic gradient that was set up by the loop of Henle. Next, we want to talk in a little bit more detail about urea and the role that urea plays in the nephron, particularly in maintaining this osmotic gradient. So we see that urea is freely filtered and it leaves the glomerulus here entering the nephron. And in the proximal convoluted tubule, we see that about half of the urea that was filtered is reabsorbed, which leaves us with 50% right here as we go into the ascending limb of the loop of Henle. As we round the bend here in the loop of Henle, we see that this 50% is joined by another 50% that, as we mentioned on the previous slide, is reabsorbed from the collecting duct. And so there's 55% reabsorbed there total from the collecting duct. 50 of it makes its way back in the loop of Henle. 5 of it makes its way back into the vasa recta and into the bloodstream. Some of that, of course, will eventually be filtered once again. So now we have 100% of the urea that we started with as we go to the ascending limb of the loop of Henle. We make our way into the distal convoluted tubule where 30% of that is reabsorbed. And then we're left with 70% as we head down into the collecting duct. And as we mentioned, 55% of that from the collecting duct is reabsorbed, meaning that only 15% of that urea is excreted from the body. It's also worth noting that this excretion is antidiuretic hormone dependent. Depending on how much ADH is released, interestingly, ADH impacts not only water reabsorption, but urea reabsorption, and so we'll have more or less reabsorbed depending on the antidiuretic hormone released. So what's the point of all this? The point is that maintaining this urea and only losing 15% of it in one pass through the nephron means that we maintain this osmotically active solute and allow it to play a role in supporting this very high concentration gradient that allows us to concentrate our urine. We want to talk next about total body sodium regulation. And of course, as we mentioned previously, that total body sodium regulation is accomplished primarily through renal function, through the kidneys. So the body has a great ability to adjust sodium excretion as intake of sodium changes. So the sodium that's excreted equals the sodium that's filtered minus the sodium that's reabsorbed. Sodium is not secreted, as we remember. So both of these variables, both the amount of sodium filtered and the amount of sodium reabsorbed, are adjustable. Sodium is the major extracellular solute, making up somewhere in the neighborhood of 90% of the extracellular solute found in plasma. And so what we see is that if we have low solute, in other words, low sodium concentrations, we have low blood pressure. As a result of this low blood pressure, we have both constriction of blood vessels and conservation of sodium. This conservation of sodium is accomplished in two ways. One is that we simply have a lower glomerular filtration rate, so we're losing less sodium per unit time. And the second is that we step up reabsorption. And so both of these factors will help bring blood pressure back within a normal range. If we look at our little diagram here, we see how this whole process works. So if we begin here at the top, we have an increased loss of sodium in water due to diarrhea. We have a decreased plasma volume, decreased venous pressure as a result of that. So first off, that decreased pressure activates the renal sympathetic nerves. And so let's pause there and go back around here. So the decreased venous pressure also decreases venous return, decreases atrial pressure, which also activates sympathetic renal nerves. And then we have this decreased end-diastolic volume, decreased stroke volume, decreased cardiac output, and decreased atrial blood pressure. So all of those are responsible for increasing the activity of renal sympathetic nerves, which leads to the constriction of afferent renal arterioles. And so when we do that, we of course decrease the amount of pressure, as you remember, that's found in the glomerular capillaries, decreasing GFR, and decreasing the amount of water and sodium that is excreted. And so it's a negative feedback loop that helps bring us back within that homeostatic range by, in this case, simply decreasing the glomerular filtration rate. We now look at the effects of aldosterone in the kidneys, one of the mechanisms by which the body regulates total sodium concentration as well as blood pressure. So aldosterone is a steroid hormone, as you know, that's released by the adrenal cortex that stimulates reabsorption by the distal convoluted tubule and the cortical collecting ducts of sodium. And the effects of aldosterone are quite pronounced. And so if aldosterone is absent, then about 2% of the filtered sodium is excreted. But if aldosterone is present in reasonable concentrations, then nearly all the sodium is absorbed. Aldosterone is relatively slow acting as a result of the fact that it's a steroid. and it induces, as you would imagine, with nearly all steroid hormones, changes in protein synthesis to exert its effects, and so all that takes some time. The basic idea by which aldosterone works is depicted in the little diagram on our screen. So the liver releases a hormone, angiotensinogen, that's 453 amino acids that circulates through the blood. And interestingly, what we notice is this, that the kidney secretes an enzyme referred to as renin. Renin is not a hormone, but rather an enzyme that cleaves an angiotensinogen into angiotensin 1, which is only 10 amino acids. Angiotensin 1 then is converted into angiotensin 2 by angiotensin-converting enzyme, which is found on the endothelium of blood vessels. And angiotensin 2 is only 8 amino acids in length, a little tiny peptide hormone. Angiotensin 2 then goes to the cardiovascular system and leads to vasoconstriction, which increases blood pressure. And it also goes to the adrenal cortex, and this is where we get back to aldosterone, which causes the release of aldosterone, which promotes salt and water retention, which also increases blood pressure. So it's evident from the previous slide that the secretion of aldosterone is controlled by the release of renin by the kidneys. The other components of that pathway, for instance, angiotensinogen, angiotensin-converting enzyme, are typically present in excess, and so it is the release of renin that controls this process. And therefore, it's interesting for us to look at what stimulates the release of renin from the kidneys. And what we see is that there are three factors that stimulate the release of renin from the kidneys. Number one, activity of renal neurons. Number two, decreased atrial pressure. And number three, decreased GFR. So let's look at the number one first. And so number one is the increased activity of renal neurons. And so these renal neurons, so if we look here, we see a decrease in plasma in this diagram on our slide, leads to an increase in the activity of renal sympathetic neurons. And so as our bullet point says there, these renal neurons impact the renal juxtaglomerular cells directly, which increases renin secretion and so forth down the line. We'll talk about these other ones next. Next, we have decreased atrial pressure. And so here we see that decreased atrial pressure also leads to an increased secretion of renin by the juxtaglomerular cells. Interestingly, the juxtaglomerular cells that themselves function as baroreceptors, which is a pressure receptor, within the kidney itself. And so that decreased atrial pressure leads to a decreased blood pressure that's detected in the kidneys, and that's what leads to renin secretion. Lastly, we have the macula densa. As you recall, the macula densa is a cluster of cells that is located in the tubule itself, and these cells detect the amount of fluid and water that's making its way past them. And so decreased glomerular filtration rate, which results in a decreased flow to the macula densa, means you have less salt and water going to the macula densa. And so the macula densa cells are connected to the juxtaglomerular cells. As we mentioned earlier, they released paracrine factors, which cause the juxtaglomerular cells to release renin. So these three level factors then trigger renin release by the kidney itself. We have an increased, the rest we've done before. We have an increased plasma concentration of renin as a result of that, increased concentration of plasma angiotensinogen 2, increased aldosterone secretion, increased plasma aldosterone, and increased sodium and water reabsorption as a result of that, and therefore less sodium and water excretion. In the last section of the chapter that we'll cover, we want to talk briefly about the stimulus that brings about the production of vasopressin, also known as antidiuretic hormone. So, as we're aware, antidiuretic hormone is released by the posterior pituitary, and interestingly, what we see is that there are osmoreceptors, in other words, receptors that actually detect the osmolarity of the blood that are located in the hypothalamus. We also remember that the hormones released by the posterior pituitary, oxytocin and vasopressin, are synthesized in the hypothalamus, so it makes sense that the receptors, these osmoreceptors, will be located there. So briefly, an increase in osmolarity, of course, an increase in osmolarity increases antidiuretic hormone secretion because there's a need to reduce the amount of water that's leaving the body and increase the amount of water in the blood, thereby decreasing the osmolarity of the blood. A decrease in the osmolarity of the blood decreases antidiuretic hormone secretion, and therefore lets more water leave the body and hopefully increases the osmolarity of the blood back to a homeostatic range. So if we look at our little diagram here, we see we have excess water ingested, the body fluid osmolarity goes down because the water concentration is going up. We have firing by these hypothalamic osmoreceptors, and we have vasopressin secretion that is inhibited as a result of this firing. We have a decreased concentration of plasma vasopressin. We have decreased permeability of the tubules, of the collecting duct portion of the tubules, to water, decreased water reabsorption, and more water that's excreted. So bringing that osmolarity back within a homeostatic range. What we'll see is this, that we're going to omit the last few pages of the chapter from 14:10 on, so this brings our discussion of the renal system to a close.